Cool, thank you. So hi everyone, my name is Maling.
Get closer to the microphone.
Sorry?
So we can get closer to the microphone.
Okay.
So we can hear you.
Yep, sounds good. Okay. So hi, my name is Maling. I'm a part of the bioinformatics services team at SOPHiA Genetics. We're the technical team that talks directly to clients about the quality and the performance of their assays with us. So today I'd like to talk to you about the challenges related to somatic variant calling in solid tumors and some strategies to overcome these challenges. Okay. So some of the variants that can be detected with RNA in solid tumors include fusion events, exon skipping events, gene expression changes in most SNPs and indels. I'll focus mostly on fusion detection because the importance of fusion genes as a biomarker is increasingly obvious in cancer diagnosis and the discovery of new gene therapies. Of course, there are challenges related to fusion detection and many aspects to consider. Do you go down an NGS route or non-NGS method? Do you look at the DNA or the RNA? There are, of course, panel design considerations, starting material considerations, etc. How you analyze the data, and then how do you interpret the results. Today, I'll dive a bit deeper into considerations related to panel design, starting material, and secondary analysis.
Just get a bit closer.
Oh, okay. Okay. So let's say you've decided to go down the next generation sequencing route and you're going to work with RNA. Great choice, by the way. There are, of course, considerations related to panel design. Particularly, look for an assay that allows you to detect both known fusions as well as novel fusions. So on one hand, you want an assay that is fine-tuned and optimized to detect a handful of well-known fusions. On the other hand, you want to look for an assay that has the capability to detect new rare fusions. So that would rule out, for example, a classical amplicon method, which would leave you with a capture-based method or an anchored multiplex PCR method. Regardless the method that you choose, there are details to consider in your panel design to detect novel fusions, particularly related to probe or primer placement. So look at the details of what is covered by the assay. So what are the fusions that will be detectable? Consider that there are different transcripts or isoforms. Be cautious of the direction of detection. So is your partner, fusion partner, a 3' or 5' gene? Consider the expression levels of the genes potentially involved in the fusion events, etc. An additional point to consider is, can your assay of choice be customized or updated in the future as you narrow your focus or expand your search? So another aspect to consider for accurate fusion detection is starting material. Library conversion rate is often overlooked, but it contributes significantly to your ability to detect fusions. So the conversion rate is the fraction of input molecules that gets converted into a sequencing library. So if you have, let's say, 10 molecules of a fusion in the sample, but you can only detect 5 in the data, then your library conversion rate is about 50%. So clearly this would affect your detection sensitivity. In the graph on top, it illustrates this point. Basically, if you can have a higher library conversion rate, then you would have more molecules detected and you would have a higher sensitivity. Another point about library conversion rate that you can see on the bottom graph, in our development of our assay, we've observed that the conversion rate is different between different fusion events. So we're still investigating the source of this variability, but just be aware that it exists, and therefore it has an effect when we talk about limit of detection. So if your library conversion rate is variable between your different fusion events, then naturally your limit of detection would also be different for different fusion events, therefore making LOD measurements quite challenging for fusion detection. So we've seen assays express LOD in terms of number of reads. However, that doesn't really account for the number of molecules that you started off with in the sample. And so we have found that quantifying LOD in terms of number of molecules measured across different gene fusion events is a much more representative way to judge the performance of a solution. So this graph here on the right is depicting the limit of detection of a number of fusion events, expressed in terms of the number of input fusion molecules that guarantees efficient detection of at least 10 fusion molecules with 95% sensitivity within a 95% confidence interval. You can see, based on the limit of detection line in the graph, that it's variable across different fusion events. And we've seen the range from about an LOD of 100 molecules to even up to 400. So the molecule count, of course, depends on your RNA quantity. FFPE samples and solid tumor samples are notoriously difficult to extract RNA from. This graph on the right here is demonstrating our ability to detect a partner-agnostic fusion in a clinical sample with relatively low tumor content, 30%. The table would indicate that we have a sensitivity of about 100% still at 5 nanograms of input RNA. So look for an assay with a low input requirement. So this needs to be taken into context. So we did work with a high quality FFPE sample. However, that's not often possible, and so RNA quantity needs to be taken into context with RNA quality. One challenge related to quality is finding a suitable measurement or an indicator of RNA quality. So we've used control gene coverage as a reliable data quality metric. It correlates well with on-target rate, which is the graph on the bottom left, and it also correlates well with read group size. So the higher the coverage, the lower the read group size, the more diverse your library. So that's a good data metric. Pre-run, we've also used DV200, which reflects the level of fragmentation that is in the sample that might impair conversion rate. And we've observed that control gene coverage moderately correlates well with DV200. The point here, though, is just make sure you have at least one quality indicator when you're running these types of assays so that you can meaningfully interpret the results. Another point is when you have a sense of quality, then you can adjust your quantity to still produce high quality libraries even if you have a poor FFPE sample. All right, so far the aspects that I've discussed are related to trying to avoid false negatives. Now I'd like to talk about some secondary analysis considerations to filter out false positives. So novel fusion detection involves complex bioinformatics. At the core, though, it involves the alignment of reads to the whole genome and then looking for split alignment, so where reads are aligning to multiple places in the genome. This concept is illustrated in the first IGV screenshot on top, which is an example of a high confidence fusion call. So you see a portion of the read that perfectly matches to, for example, NCOA4 exon 8, flanked by soft clip sequences. If we blasted that soft clip sequence, it would match RET exon 12. And then when you go to RET exon 12, you see a corresponding pattern of a portion that maps perfectly in soft clip reads. So this would be a typical pattern and amount of coverage that would represent a true fusion. Interestingly, the IGV screenshot on the bottom would actually initially also be called a high confidence fusion. So in one region of the genome, you see two supporting reads that align well, and then in another corresponding place in the genome, you see high coverage. However, this high coverage is actually due to many poorly aligned reads with few supporting reads, actually just one. And so the challenge with secondary analysis and novel fusion detection is how do you differentiate real signal, real fusion events from the noise? And so a number of filtering strategies needs to be implemented in the pipeline of choice. Certain filtering strategies can be related to pipeline parameters, so setting a threshold for the number of unique molecule count for a real fusion versus noise, or setting a threshold for read support or statistical significance, so the probability that the event is a real fusion. But there are also nuanced filtering strategies that you can employ, for example, looking at genomic context. So where's the location of the breakpoint of the event? Is it in the exon or intron? And related to that, what is the coding significance of the event? Most clinically relevant fusions, their junction point is exon flanking, so you can filter a lot of false positives just by excluding intronic events. Similarly, are these events found in homologous gene regions, in pseudogenes, etc.? The point is, the pipeline needs to have filtering mechanisms to determine the high confidence fusion calls from the candidates. Another filtering strategy that we've seen being used is to look at expression imbalance. Expression imbalance is basically when the first few exons of a highly expressed gene B is fused to a lowly expressed gene A, and so gene B is driving the expression of exon 4, 5, 6 of a lowly expressing gene. And then in gene A, you'd see a change in coverage in the later exons. So this would indicate that there's a fusion in exon 4, involving exon 4, 5, and 6 of gene A, and it would also allow you to predict the breakpoint. However, there are limitations to looking at, to using expression imbalance for novel fusion detection, because one, you can't identify what gene B would be. As well, sometimes imbalance is difficult to detect. So in the two first graphs on top, this is a clear case where imbalance can support the fusion call. You see a clear change in expression, and both of these are real fusion events. However, there are cases where the imbalance is just a result of noise or, and not related to a real fusion, or as the bottom graph shows, a real fusion may not actually exhibit any change in expression. So we've seen assays where expression imbalance is used as the primary source to detect a novel fusion. However, we find that it's just useful information, particularly in borderline cases where you want more supporting evidence for a real fusion. So taking a step away from talking about fusion detection, you can also use RNA for SNP and indel calling, and the ability to detect SNPs and indels in RNA sequencing data has been explored in academic research over the last decade, and you see many, many publications on this point. One of the biggest benefits is in certain applications, it might be possible to run just an RNA-only workflow. However, there are particular limitations to this. There are cases where it's impossible to do SNP and indel calling in RNA, particularly if your region of interest is not actually expressed in your cancer type of interest or your tissue of interest. Additionally, if you're looking at SNPs or indels in intronic regions or in promoters, that's not possible with RNA. So in these cases, combining your RNA and DNA analyses may be the most appropriate approach. You would get the versatility of being able to detect multiple biomarkers, and you get the flexibility of how you want to run the workflow. And of course, this all depends on the specific question that you're asking, and turnaround time, etc., other considerations. The takeaway point here is there are many options available for you to tailor the workflow to your particular needs. So with that, I'd like to direct you to Mikhail, who will talk to you a bit more about our own RNA target solution.
Thank you, Mei-Ling. And hello, everyone. First of all, it's super exciting to be here in person. You know, it's an incredible feeling to stand in front of the people instead of staring at a camera. But my name is Mikhail Pertziger, and I'm a product manager for SOPHiA Genetics. And Miling did all the heavy lifting. I'm just really to wrap it up. And what I wanted to do is to really introduce, as Miling said, the product that we've just launched, SOPHiA DDM RNA target technology. And before I jump into the depth of my 2 slides that I have, I just wanted to start off with a question, and that is, when you are onboarding a fusion detection assay in your lab, what sort of compromises do you have to make or do you think of, right? And while you are thinking about this, one of the typical compromises that we hear of is the labs really have to make a decision whether they want to detect novel fusions on one hand, or whether they want to work with low sample input, right? And these seem to be the 2 things that cannot be brought into one assay. And I wanted to mention the fact that this was at the top of the agenda when we were developing the product. How do we provide the labs with the benefit of the novel fusion detection, which is absolutely becoming more and more essential in the current day and age, right? But also being able to work with very small samples, for example, lung biopsies, where you've got very tiny samples and you want to do a lot of assays on them. So in the end, I'm not going to go through all the other benefits of the solution. I just wanted to mention that it's a solution that really brings an end-to-end workflow that really maximizes the insights from small biopsy samples. There are a lot of the things that I think Miling has mentioned in her talk, and there's a lot of considerations that we have put into this assay, and I'm not going to go through them. But what I would encourage you to do is, if you have any questions about RNA, about DNA, anything NGS, or you just want to have a chat, come and see us at booth number 2. We are right at the entrance there, and we would be more than happy to answer them there. But if you have any questions right now, I think we might still have a couple of minutes to address them now. But yeah, as I said, just 2 slides.